Okay.
Let me introduce you, and then you can start. Dr. Ford is one of the most distinguished figures in our field. She's a pioneer whose work has been fundamental in using translational neuroscience to understand why people hear voices. We are privileged to have here closing this session with her lecture, Hallucinations in Psychosis, Vulnerability, a Translational Neuroscience Approach. Okay, Dr. Ford, the floor is yours.
Great. If you can't hear me,
yeah, perfectly.
You can hear okay?
Yeah, perfect.
And see. Okay, perfect. Thank you for this opportunity. I wish I could be there. I love Spain, and I would love to be there right now. And also, I appreciate this the last speaker berth, because a lot of what I was going to say has already been said. So this will be a good review for people who have been here the whole time, and maybe new for people who haven't been. I'm going to start off with an outline of what I want to talk about. I'm going to talk about neurophysiological studies of the corollary discharge efference copy system in healthy humans and non-human animals. I'm going to focus on corollary discharge and the efference copy. I'm going to talk about neurophysiological studies on people on the psychosis spectrum, and then any relationships with symptoms of psychosis. Again, we make a distinction between corollary discharge and efference copy, and I think Anna Pinheiro, Tom have both, and maybe Rosa have made it clear that there is a way to distinguish them. So let me just start with this. This is something that I think you've all seen one form or another today. This would be the nematode. This would be the human primate who can't tickle himself. And then this was also some of the high primate data that have been shown. We haven't talked about songbirds, but we know that this system has been studied across the entire animal kingdom. Any species that can move needs this system. It allows them to predict and thereby suppress sensations that result from their own motor actions, and then tag them as coming from self. It is a system that fosters economical processing by allowing external and behavioral relevant stimuli to have priority. I'm going to show our model. I think there have been a couple slides showing this. I usually show Anna Pinheiro's model because it is so easy, so much easier and more elegant, but this is kind of what I'm working with. I also want to highlight that I have added the cerebellum to the way I think about things. Since I've been doing EEG studies since 1970, by the way, I'll help you with the math, that was 56 years ago. I'm very, very EEG focused, and the cerebellum is not easy to record EEG from, so it has been largely deleted from the way I've been thinking about things until now. And now I understand its incredible role in this system. So yes, I have been corticocentric, but I'm trying to change that. So here's sort of the way we think about it. I'm just going to give some basic anatomy. That's the auditory cortex. That's the thalamus. That's the pons. And here's the cerebellum. And in our paradigm, which you've heard a lot about, the talk-listen paradigm, people say, ah, and there's an intention to speak. And that intention is sent to motor cortex as well as supplementary motor cortex, and the action happens. The person says, ah, it goes through a microphone into an amplifier system and into the ear. At that point, we postulate that it goes to the thalamus first, and then it goes to the cerebellum, and a representation of that sound is deposited in the cerebellum. Now, if I were better at PowerPoint, I would have been able to make this simultaneously happen. That would be the efference copy from the motor command would have simultaneously appeared here in the pons, going to the cerebellum, and then a representation of the corollary discharge would be deposited also in the cerebellum. And as has been described, if there's a match, then the higher cortical regions are minimally involved. But when the sound doesn't match, responses are not suppressed and higher cortical regions are likely engaged. Temporal lobe and also even the frontal lobe. This corollary discharge is basically the sensory side of this whole model. And that's what I'm going to start talking about right now. We were inspired by the monkey work. Initially, Miller-Pruess and Plug did these studies in squirrel monkeys. Squirrel monkeys do not naturally vocalize. They're not as social as marmosets. Later studies by Eliadis and Wang used marmosets who engage in vocalizations, communication, sort of a call and response kind of species. And what I circled here are the spectrograms of when their vocalizations occur. And they're loud scree sounds. And you can see that in this plot right here, these are auditory neurons. They're going along, they're firing, but as soon as the monkey makes one of these sounds, it's suppressed. Importantly, not all of the cortical neurons are suppressed. Some of them are excited. But during vocalization, most of the neurons in auditory cortex are suppressed. These people counted them. They counted the neurons, and 75% were suppressed. But some are excited, as you might expect, because you actually do hear what you say. Also, possibly important is the fact that suppression occurs before vocalization onset. And this may have something to do with the readiness potential that has been discussed already and that I will discuss later myself. So that was monkeys vocalizing. Here we go with humans vocalizing. Here's our guy with an electrode cap on. So as I said, the non-human primate studies guided our approach to studying the efferent copy corollary discharge mechanism. And like them, we used electrophysiology recordings from auditory cortex during vocalization. And we used EEG-based methods. Here's how we do it. This is an old slide, but I couldn't resist it. Our people say ah, 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 ah. There's a reason they say ah, and that's because it disturbs the least number of structures in the head. The jaw is less involved, the tongue, the throat, etc. And we aren't the only people to use ah. So we discovered it, but a lot of other people who were doing similar work also started using ah. And the event-related potentials, or ERPs, are derived by averaging together the EEG segments, time-locked to voice onset during talking. We refer to this as talking. It's really vocalization, but it just is too long to say that every time. So we refer to this as talking. And this is our talk-listen paradigm. And the speech is recorded during talking and played back during listening. And ERPs to voice onset are compared during talking and listening. And of course, we predicted suppression of neural responses during talking compared to listening, consistent with human and non-human primate literature. This is something I think you may have seen, or some version of it. This may have been from our first paper. It is an event-related potential, or ERP, and the N1 is the first large negative component of the ERP coming from auditory cortex. As a field, it took us a while to agree that it actually comes from auditory cortex, but I think it's pretty solidly established at this point. N1 to speech sounds during talking is suppressed relative to the N1 sounds during passive listening, that is, of the speech played back. And this is a suppression of N1. It's our assay of the corollary discharge. And as I said, this has been attributed to the corollary discharge mechanism. A little black box warning here. N1 is very sensitive to the physical parameters of the sound, such as intensity, rise time, even the duration and the interstimulus interval. Therefore, N1 suppression during talking must be calculated relative relative to N1 during listening to the exact same sounds in the exact same sequence. I just want to show you a study we were lucky enough to do when I was at Yale with a neurosurgeon, Dennis Spencer. These data were recorded from patients who had intractable epilepsy, and they were recording from the cortex of these patients to both find out where the source of the epileptic seizure was and to find areas of the brain that were considered eloquent cortex, that is, cortex involved in speaking and listening to speech. This is a grid over that whole area of the brain from one of the patients, and you can see that the biggest suppression, again, same thing I just showed it a second ago, this is listening to the playback of the patient's awe, and this is during talking. The patient is saying awe, and it's biggest there over at that electrode. We also were able to look in some patients at what's going on at what we considered might be Broca's area right there and auditory cortex right there. And again, this activity was greater during talking and Broca's area than listening. And this has already been shown. This was something that Jun Wang did in our lab some years ago now. He did a source localization of the activity, and the red is what was greater during talking than listening, and that's during the pre-speech onset period. So we sort of thought this is probably coming from Broca's area and the right hemisphere analog of Broca's area. On the other hand, we have the green areas. This is the superior temporal gyrus, where listening was greater than talking during the post-speech onset period. And this was lovely confirmation, both of the data we collected during the surgical evaluation of patients for epilepsy surgery, and this is all in healthy controls. There was significantly more phase locking between frontal and temporal sources, both in the delta and theta bands. So this is a kind of neural synchrony. These areas became synchronous, more synchronous during talking than during listening. And furthermore, the degree of synchrony between Broca's area and Wernicke's area, if you will, predicted the amount of cortical suppression. Here's another study we did a while ago. We were looking at the synchrony between frontal and temporal areas during talking and listening. This was actually an interesting study sort of in the history of our work. This was before we hit on the idea of recording the brain's response to awe. These were people saying sentences. We gave them pre-written sentences, and we asked them to say these sentences. While they were saying these sentences, we recorded the EEG, and we found that the synchrony between the frontal areas and the temporal areas were much greater during talking than during listening. And some of these lines are significant at the 0.001 level. So it's important in this area to be able to study animals who don't vocalize. So marmosets vocalize, humans vocalize, but a lot of lab animals that people want to be able to study don't vocalize. I actually consulted with this group. This was Torfi Sigurdsson, and he's from the Rummel group, about maybe how to study this in rodents. And remarkably, I think, they taught rodents to press a button to hear a sound. The rats or rodents are pressing a button to deliver a sound, and also there are random sounds being played during the same session. And I'll just skip to the punchline here. These are recordings from auditory cortex during the listening condition, that is here during the random condition with the blue, and the red here is the recordings from cortex when the rodent was pressing the button. So you can see there's much more activity in auditory cortex during listening than during pressing. So we also have done press a button to hear a tone, and I think many of you have seen those data already today. Basically, we taught humans how to press a button to deliver a tone, just like the other group taught rodents. So you press a button, you hear a tone, and you see N1 suppression. I think it's interesting, and we have a paper on this, comparing the N1 suppression during pressing a button to get a tone versus pressing a button to hear your own ah played. There is suppression, obviously, here in N1 during the button press task, but there's much more suppression during the talking task. And it's probably because of the overlearning throughout your entire life that you have learned to associate talking with that sound. There's also an interesting animal literature on precision of the sound. So here again, this is data from another study by Eliadis and Wang, and they had they distorted the feedback while the monkeys were vocalizing. They were able to pitch it down, and this is a plot of the response, the firing rate. This is when the blue line is when there's no altered feedback. This is just the regular condition. The monkey is making a scree sound, and he's got a lot of suppression when he's doing it himself. But when they introduce an altered feedback, so it's not what the monkey said, it's a little different, the suppression is much less. We did a similar thing with Tata Hanks Maldonado. She also did a feedback distortion. So this is the N1 during talking that you've seen before. And this is, this is just normal feedback. The people are just saying, ah, ah, ah. But then some of the times the feedback is a little different and it's pitched up. So it sounds kind of instead of and there is less suppression when the sound has been distorted. It is not what you said, and you do not suppress the response as much. Obviously, it's something you need to attend to. It probably gets a message sent up to the temporal lobes, obviously auditory cortex, but undoubtedly frontal lobes as well. So that's been sort of the sensory side. That's the N1 side of the story. There's also the efferents copy motor side. And let me just remind you that that's this big red line going straight from motor cortex down to pons. I'll explain that later. And thalamus and cerebellum. So what happens when, let's get the motor system involved here. This is a beautiful study by the Schneider and Mooney group. This is by Anders Nelson. They were able to use in vivo intracellular physiology, optogenetics, and sound playback. They found evidence directly that activating supplementary motor axon terminals in auditory cortex. So they were able to find axon terminals in auditory cortex that came from supplementary motor area, and they found that that when they stimulated supplementary motor cortex, those auditory cortex was suppressed compared to when there was no motor stimulation. So when they did not stimulate SMA, auditory cortex was not suppressed, but when they did, it was suppressed. So how do we study that? We can't do that. So we're doing sort of the what we were hearing everybody else talk about, sort of the readiness potential. Now we're looking at what happens right before you speak. This is an early paper that we used time frequency analysis with. And we, again, looked at synchrony from trial to trial. So in this example, and I made this up, each little box of EEG is in phase with all the other trials. And this measure reflects the synchrony of neural activity across trials. It can occur in any frequency band. In our case, we couldn't look lower than beta because of the interstimulus interval of our event. So we were restricted, but we found more synchrony from trial to trial to trial during talking than during listening. And here we have speech onset, we have time. This is about 100 milliseconds before speech onset, 200 milliseconds before speech onset, and this is from healthy controls. And you can see that there is more synchrony from trial to trial during talking than there is during listening. I've said that. And we asked whether it correlated with the N1 suppression that followed that. And indeed it did. This is a plot showing the amount of pre-talking synchrony related to the amount of suppression. So the more synchrony, the more suppression. And this is similar to the rodent studies from the Schneider and Mooney lab. Like the efference copy preceding talking, there's an efference copy preceding the button press to deliver a tone. And it is also related to suppression of the N1 to the tone. I think you've already heard a lot about that. This is just a paper a while ago that we published in, I think, Schizophrenia Bulletin. And it relates more suppression with bigger LRPs. You've heard about the readiness potential. This is a lateralized readiness potential. So when you press with your right hand, you have more negativity over left cortex. When you press with your left hand, you have more on the right, and it's considered lateralized. So the larger the preparation basically to press that button, the more suppression you have. So there it is. Subjects with greater N1 suppression had greater lateralized neural activity preceding the press that delivered the tones. Okay, let's get to the meat of this talk. This is how do neurophysiological studies of corollary discharge in people on the schizophrenia spectrum. I did a little search. I don't know whether I did this correctly. This seems a little bit too impressive. I put schizophrenia and corollary discharge or efference copy into my search engine in PubMed, and I found out that a couple days ago that 762 papers came up. That's a lot. So we're not the only ones doing this, folks. I want to quickly show you, again, because I'm trying to show how all of what we do with humans can be related to non-human species. This is that same group, the Torfey-Sigurdsson group here, Rummel and Sigurdsson, and this is the same paradigm. But this is interesting because this is a mouse that's been genetically engineered to have the 22q11 microdeletion. And in humans, the 22q11 microdeletion is one of the largest genetic risk factors for schizophrenia. And what you see in this picture is that they found that auditory cortical neurons fail to attenuate their responses to self-generated sounds, recapitulating the deficits seen in schizophrenia patients. So here you see the self-generated ones. The wild type have suppression, but you can see random and self-generated, there's no difference, but there is in the wild type. So auditory cortex displayed weaker motor-related signals and received fewer inputs from motor cortex, suggesting an anatomical basis for deficit in sensory suppression in schizophrenia. And this appeared a couple years ago in Nature Communications. But what about humans with schizophrenia? That was a mouse with schizophrenia. Here are humans with schizophrenia. Again, you've seen this. This was, I think, from one of our first papers. It was healthy controls. Somebody asked about the difference between FC, FCZ, and CZ. I see here that we chose CZ for this particular, in this paper. And I know it changes actually slightly. The effect sizes are a little bit different using the different electrodes. So this is the N1 suppression, the operation of the corollary discharge, and is in effect. And just sort of within our own group, we originally did this at Stanford, at the Palo Alto VA. Then we moved to Yale and did this at the West Haven VA. And then we moved to San Francisco, and we did it at the San Francisco VA. And then we worked together with Tom Whitford's group in Australia, and then colleagues at the University of Illinois, Chicago. So we get it East Coast, West Coast, Northern Hemisphere, Southern Hemisphere, and the middle of the United States. And now in Spain, which is terrific. I love seeing replication. That's one of the reasons we published that paper, that in the Nature Methods paper. We hoped other people would pick it up and do it and see if they get the same thing we get. This is when people are pressing a button to get a tone. We see the corollary discharge deficit in schizophrenia seen with the button press paradigm. So that's healthy controls. Those are people with schizophrenia. I think we already saw that today. And then another question that came up today too, is it specific to schizophrenia? I don't know why I thought it would be, but I did, thought it would be. And our colleague, John Sweeney, who passed away a couple years ago, sadly, ran this study for us in Chicago. And it is seen in people with schizoaffective disorder, psychotic bipolar disorder, and first-degree relatives to an intermediate extent. At least we see it in bipolar. I know, Rosa, yours is a little bit of an intermediate finding, but possibly reflective of a vulnerability for psychotic experiences. Also, again, I think Tom showed this. There's a corollary discharge deficit is seen in clinical high risk for schizophrenia. It's also seen early in the illness course. This is from this is a big sample for us, and you can see that the clinical high risk population also has more of a schizophrenia looking pattern than the healthy controls. We also, because we were at Yale and they do these ketamine studies, we were able to give ketamine to healthy volunteers. There's a glutamate receptor hypothesis of schizophrenia, that schizophrenia has hypofunction of the glutamate receptor. And so we gave people ketamine, which is an antagonist of glutamate. And what you see here, and at the same time, we also studied people's schizophrenia. Just sort of look at the top line. This is FC. You can see healthy controls had what we expected, which is suppression of N1. People's schizophrenia had less suppression of N1. And then healthy controls, we gave placebo and ketamine. And you can see that ketamine recapitulates and mimics some of the features of schizophrenia. So we refer to this as schizophrenia in a bottle, which is actually not quite true. So what is the relationship of all this with symptoms? One of the things I didn't tell you as we went through all the different studies was that the N1 suppression effects that we show, East Coast, West Coast, middle of the country, none of those correlated with the symptoms of interest. Smaller suppression did not correlate with auditory hallucinations, which is, obviously very disappointing, because that's the reason we got into this field, was to understand this particular symptom of schizophrenia. I'm just going to throw out a few things that did work. This is in that clinical high risk sample, the CHRs, the clinical high risk youth. The worse suppression they have, the higher their delusional thinking. It's called unusual thought content in the high risk group. So that's good. That suggests that there is something about the psychosis experience. Maybe these relate to like the anomalous self experiences, and the more these people had, the less suppression of N1 they had. I showed you this already, sort of. This was Tata Heink's study. This is where healthy controls have normal N1 suppression when there's just regular old feedback. But this is when we altered the feedback, they had less suppression. And we found out that this effect is also seen in non-hallucinating patients. So these are people with schizophrenia who don't hallucinate. So you see it in controls and you see it in the people who don't hallucinate. But you don't see that effect in the hallucinating patients. So that's one little hook we have to the hallucinations symptom. Another one is the study I showed a minute ago, which was the neural activity preceding talking. People with schizophrenia, you can see, don't have much by way of neural synchrony preceding the talking as the healthy controls do. So we're asking whether this is related possibly to auditory hallucinations. And it was. This is one of our successes. Pre-talking synchrony is reduced in patients with a propensity towards severe auditory hallucinations. And this is corrected, by the way, for other kinds of symptoms that people with schizophrenia have, like thought disorder, negative symptoms. And this you might recognize from a few minutes ago. We also did this in people with schizophrenia, and I showed you that there's a lot of neural synchrony between frontal and temporal lobes when you're talking in healthy controls, but we don't see this in patients. Kind of remarkably missing, I would say. And we find that that actual missingness is probably driven by the people with schizophrenia who hear voices. So there's some difference in the people who don't hallucinate, but there is no difference between talking and listening in the people who do hallucinate. So this is another hook into hallucinations, may have something to do with the motor activity, the message being sent from frontal lobes to temporal lobes is broken and is possibly related to auditory hallucinations. Where's the cerebellum? Yes, good. I'm finally getting to the cerebellum. Like I said, been very corticocentric because that's what EEG records. But I have learned from colleagues, I think Anna, one of them, has put cerebellum on my radar, and we've begun to look at it. Also, here you go. I also did the same search. There have been 118 papers on cerebellum and corollary discharge or efferents copy. Again, I don't know if we can trust these results, but this is what PubMed has spit out. This is a study that came out, probably one of the first studies. This is, I think Anna may have mentioned this study. This is from the Knoll group with Sonia Kotz, Eric Schroger, and Pamela Bess. And they were studying people with cerebellar lesions. And by the way, negative voltages are plotted up here. If you've been trying to get a grip on what I've been talking about, it's usually going the other direction. But you can see here, healthy controls, you see a lot of suppression. This is N1, even though it's going up. There's a lot of suppression in healthy controls, and there's less N1 suppression in people with cerebellar lesions. Which brings me to my final point, which is a study by my young colleagues, young, wonderful, smart colleagues, Sam Abram and Jessica Waugh. They put together data from the Chicago study that I mentioned that John Sweeney led, and were able to correlate connectivity between the pons and the cerebellum and N1 suppression and hallucinations in schizophrenia. This paper came out last year, maybe 2 years ago, in Biological Psychiatry, CNNI. And this is sort of jumping straight to the final, most impressive picture. And this is the talk, listen amplitude. This is suppression on this axis, and this is pons to cerebellar vermis connectivity. And you can see that the greater the connectivity, the more suppression. This is in unaffected comparison participants or healthy controls. And this is in people with schizophrenia. And you can see, it did not work in people with schizophrenia. There was no connectivity relationship and to N1 suppression in people with schizophrenia. However, there was a relationship to the PANS P3 item on hallucinations. So people with greater hallucinations had less connectivity between the pons and the cerebellum, the vermis. I am not a scholar of the cerebellum, and I wish I were. I know there are a lot of parts of it. Anna, I think, did a wonderful job of showing all those data. The vermis is apparently involved in motor control, especially of the head. Maybe this is just all about talking and head movements or not. But the nice thing about the vermis is I think it's a pretty easy place to stimulate, and we think it has important treatment implications. In fact, non-invasive vermis neurostimulation has been related to reduced negative and depressive symptoms and increased frontal cerebellar connectivity in people with schizophrenia. And because frontal cortical regions are connected to the cerebellum via the pons, we speculate whether vermis stimulation could enhance pons to cerebellar vermis connectivity in schizophrenia and in turn improve sensory prediction and hallucination severity. Okay, I'm going to stop there and summarize. What have I learned? We had hoped to understand the pathophysiology of voices by assaying the corollary discharge efference copy mechanism. I would say we had only partial success. Basically, the abnormal corollary discharge, that is to say the N1 suppression, did not really map onto voices. Abnormal efference copies may be better at mapping onto voices, but evidence is so far limited. And I'm very heartened to know that you people here are working on this. Instead, what do we have? We have deficits in the corollary discharge may signal psychosis vulnerability because it is seen in psychotic bipolar disorder and possibly to a lesser extent than in schizophrenia. It's seen in clinical high-risk youth, especially in delusional youth. And to some extent, it's seen in first-degree relatives. Tom Whitford's group has shown that it's seen in schizotypy, which may relate to what we've been hearing about already about maybe non-clinical voice hearers, people who hear voices but don't get clinical treatment for it. That may be embedded in the schizotypy findings of Tom Whitford's group. And we also see it in a glutamate model of schizophrenia. So I haven't lost all hope. I think that there's hope in this N1 suppression game. And one of the things that I found, we're also going to follow our leads with cerebellum and pons, but I found Dr. Madier's comments very interesting. Hallucinations are difficult to assess, and they're not always assessed as well as they could be. They're assessed by carefully trained research assistants in some cases. Really depends on how you ask the question. People can be very guarded about it. You have to, as a colleague I worked with at Stanford for years, said, you have to give people time to leak psychoticism. So you can't just say, do you hear voices, and trust that answer. You have to have a relationship with them. You have to sit with them. You have to listen to them. And then maybe you get the answer. With that, let me point to colleagues and funding that I've had and welcome questions. Let's see.
Thank you, Judith. Maybe you have some questions.
Let's see, how do I stop sharing? Oh, there. Oh,
hello. Can you hear me? Hello.
Hello.
Hello, can you hear me?
I can hear you.
Oh, okay. Okay.
I can't see you, but I can hear you.
I think we have some questions here for you.
Okay, great.
Antonio.
Hello, Judith.
Hello.
I am Antonio Arjona, fan of your work.
Thank you. Thank you for your talk, by the way.
You're welcome.
And the readiness potential. Very nice to have that explained.
Yeah, I had a short time. I have a lot of questions, so I'm going to ask one of them. When we talk about pre-speech pre-speech preparation, we are talking about frontotemporal connectivity or coherence. Mm-hmm. I've been reading about this, and my conclusion was that the theta band was the most accurate band related to this process. But I see that you have one study which talk about the beta band. I'm not sure. Do you think both are equally useful for this process analysis, or these are different ways to analyze this connectivity?
I am a bigger fan of theta than beta. The reason that we analyzed the beta band was because of the limitations of the data we had collected. Our interstimulus interval was too short. We could only look at higher frequencies, unfortunately. But theta band has worked better for us in other studies.
Okay.
So in the study that the Wang et al. Study, that was a theta band finding, delta and theta. In the paper that showed during our talking in sentences, that was theta band.
Okay, so we'll keep looking at the.
Yes.
And another question. We are now talking about the cerebellum.
Mm-hmm.
This implication is about the movement that you make when you talk, or about the sense of, the hearing of the voice. Which part is the function of the cerebellum in this processing?
We actually found vermis was the one that had the strongest, most significant connectivity. But we also found things in, I think, crust 6 and 7. I know most about the vermis because that's where the significant findings were, and I don't know. It's I need to learn more about the cerebellum, and I'm counting on young, smart people like you to lead the way. I don't know why the vermis worked out.
Yeah. Okay, we'll work on that.
You work. Thank you.
Thank you very much for your talk.
Thank you.
Any other question?
This is Vicente Molina. Just a very quick question. What method did you use to measure the connectivity between pons and cerebellum?
It was resting state connectivity from the study conducted in Chicago, and it was basically looking at a correlation between the bold response in the pons, the bold response in the cerebellum, everywhere in the cerebellum, everywhere in the pons, and everywhere in the thalamus. So it was we expected to see something in the thalamus, but we did not. So it was really just your standard resting state functional connectivity of resting state data. I think it was 6 minutes. I think it was 6 minutes eyes open, but I'd have to check to let you know. I can look right now if you want to know. Let me see.
Okay, thank you. I think we have some questions in the chat. Maybe they can speak for themselves better than reading the question. Joao Pereira is first. Can you speak?
Yeah, can you hear me?
Yep. Yeah, perfect.
Okay, thanks. So yeah, thanks for the presentation. I was wondering, so when we hear the recording of our own voice, almost everybody has this strange sensation that it doesn't sound exactly like the voice we hear while we are speaking. And I was wondering if you ever accounted for this mismatch in this speaking listening paradigm.
Thank you for that. You're right, it doesn't. So we do pitch it. I think we pitch it down, and so that it is. I do believe. I'm trying to remember. I think that the skull distorts the voice down about 3 hertz. Is it a semi? It's a semitone. So what would that be? I can't remember, but we do try to adjust for that. Yeah, I should have mentioned that, because it does sound, your voice does sound different when you hear it back. It sounds higher than what you think you sound like because of the bone, the skull changing the frequency of it. I just figured out how to turn my camera on, folks.
Thank you.
I mean, I think that's important. And so in the study that Theda Heinz did, she pitched it down, and then she pitched it down another semitone to distort the voice, so that it sounded even more distorted. Also, there's another study that I didn't have time to present that I really like. It was not different. It was not affected by schizophrenia and certainly not by voices. And that was a study that an analysis that Kevin Seatek did, where he looked at the format of every single speech sound, and when there was a deviation from the previous speech sound, he saw that there was less suppression than the sounds that were not different from the previous speech sound. So that he had like 120 ahs, and he was looking at each and every one of them, and when they were more different from the preceding one, there was less suppression than when they were more identical to the preceding one. And
I think I have, if I may now ask a question. Hey, Judy, thank you so much for your fantastic talk. It's really nice.
Thank you for your fantastic talk.
It would be much better if we were presentially both in Spain. I have a question, and what I would like to ask you is whether, based on your findings and also, well, some of the findings I have presented today, can we say that auditory hallucinations are somehow the price humans pay for speaking? And in your view, why is the voice system particularly vulnerable to what we could describe as atypical perceptual experiences? I have been thinking a lot about that, and I would love to know your thoughts. Thank you so much again.
Yeah, thank you, Anna. And like I said, thank you for your great talk. Yeah. So, I've thought a lot about hallucinations, as I'm sure you have also, and I could go on and on about how I'm not really so sure that they are an inner speech phenomenon. I often think that they are just a sense of communication, a sense of meaning, and is language necessary for that? Yeah, maybe it is. But they do have anomalous self-experiences in other domains also. Certainly there's some people report having a feeling that someone's tapping them on the shoulder. Or, I mean, it's rare, but they will have the olfactory. And then another hallucinatory experience is visual hallucinations, which I think is much underappreciated in the field and generally sort of ascribed to over-endorsing. The patient is just over-endorsing odd experiences. But we had data from a while ago showing that people with visual hallucinations also have aberrant connectivity, in this case between visual cortex and the amygdala, as you might expect. And I wish that we had done your scale on anomalous self experiences, and I feel like that is possibly easier for people to endorse and understand than auditory hallucinations. Yeah, I don't know. What do you think?
Well, yeah, it's I guess that there is, well, at least that's what I tried to show today, that there is more to the voice than the sensory motor circuits that we have been interested in characterizing, and even some aspects, some communicative aspects of the hallucinations themselves. So I guess that we also have to think that behind a voice there is a speaker. So all these social trait inferences or speaker identity perception, but it's true that of course they can occur in other modalities, but perhaps the voice is somehow unique in that sense that it could, yes, represent both communication. So behind the voice there is a speaker and all the aspects related to speaker perception, but also these self dimensions that we have been referring today. and that could make this system particularly vulnerable, and therefore accounting for the complex phenomenology of voice hearing. But there is certainly more studies that we need to conduct in order to understand that complexity for sure.
When again, reflecting on Dr. Madeira's talk on aspects of it, I have really valued talking to patients and actually hearing about their experiences and how they think this happens, and it's quite varied, I have to say, and not everybody can identify it with a human being. Not everybody knows, even once they know that it's from a person, they can't identify who it's from. It's it has social implications. It's, I don't know, we could go on and on talking about the phenomenology of auditory hallucinations, and in our work we don't really touch on it as much as I think we should. I think that if we broke it down, we might have a better run at it.
Yeah, for sure.
But thank you for raising the question of
thank you so much. Thank you.
And another thing to think about is thinking. I've spent some amount of time thinking about thinking. Do you think in sentences? I mean, I know I can think in sentences. I can prepare to say something and get it out, and it's in my brain in a sentence. But as you walk through your house, as you walk to work, are you thinking in sentences? Are you thinking in words?
That's a good question.
I don't know. It gets very difficult to think about.
Yeah, for sure.
Okay, I think we are done now.
You guys can go home.
We are
go to bed.
Here in Spain. It's 8 p.m. and I think we deserve to rest.
Do people in Valladolid eat late like they do in Madrid?
Sorry?
Do you eat late in where you are?
We didn't have dinner yet.
Okay, good. Okay.
So we are starving. We are also
okay. Thank you for the opportunity to talk to you.
We are delighted to having you here joining us today. So you're welcome to come here to Spain whenever you want.
Okay, I will. Thank you very much.
Thank you.
Okay. Bye-bye.
Bye-bye. So we are closing now the conferences. I think we are leaving home now. Thank you.